Here we are. So hi everyone, I'm Yue Qin. I'm a PhD student from Editor Lab. Today I'll be also sharing the current and future status of our recently published work, which please check out our paper if you're interested. It basically gives you a method for mapping cell structure across a variety of scales by fusing protein images and interactions. So today is a joint presentation with Leah, if she is here on the stage.
I think I'm back.
So Leah will could you please help introduce the project?
Yeah, definitely. Hi everyone, I'm Leah Schaffer. I'm a postdoc in the lab. So cells form this multi-scale structure with proteins participating with one another in protein complexes. These protein complexes interacting and participating in these larger biological pathways, and then these pathways occurring within and between different organelles that then make up the cell as a whole. But how proteins form the cell is still largely unknown, and one of the major goals as biologists and proteomicists is trying to determine this multi-scale structure. Next slide. So one of the maybe most well-known examples we have of one of these maps of cell structure is the Gene Ontology, where larger Go terms at the top of the Go hierarchy, such as the cytoplasm and the nucleus, are factored into smaller and smaller Go terms that then will describe, protein complexes or subunits of protein complexes. But however, although the Go is a great resource, it's still largely incomplete. There's many proteins that are not well annotated in Go, so they might be only annotated to, the cell term or much larger terms at the top, but the smaller protein complexes or pathways they participate in is unknown. And then there's also still many largely unknown complexes and functions for many proteins in the proteome. Next slide. So one active area of research in the IDCR lab is trying to create maps of cell structure derived systematically from data. For example, starting with gene or protein similarity networks, then using these as the basis for systematically converting them into hierarchical models that describe cell structure. And so two of the major modes we have for characterizing cellular components are protein imaging and interaction data sets. Next slide. And so for protein interactions, for example, affinity purification mass spectrometry, a bait protein will be pulled down and the praise will be identified using mass spectrometry. And then you can create these protein interaction networks like shown on the right, drawing an edge between these bait-prey interactions that are identified. And through this, you can see in these networks, then you can kind of see these larger structures forming that, represent different protein complexes or even subunits of complexes. And then on the other hand, there's this orthogonal approach of protein imaging data where a protein of interest will be stained for with an antibody conjugated to a fluorophore. And then other different cellular markers, such as the nucleus or microtubules, can be stained with other fluorophores so that in the final image, you're able to then spatially map out where the protein is in the cell relative to these other cellular markers. And so these large-scale imaging and interaction data sets in recent years, there's been a push to, try to systematically create these large-scale data sets. So for example, on the left, we have the BioPlex initiative with the Gigi lab at Harvard, which has been systematically performing APMS experiments. So this is actually from the very first BioPlex paper, but there's even more interactions now, but performing thousands of different APMS experiments to create these global protein interaction networks. And then on the right, you can see, at the same time with the Human Protein Atlas has been systematically performing imaging experiments. So collecting these confocal images with antibodies of different proteins. It's become a resource with over 10,000 different proteins with images collected. Then you can see then they are trying to determine the subcellular locations of proteins. So for these thousands of proteins, the subcellular location has been determined. And so, yeah, the question that to be answered still was how best to integrate these major resources we have in order to build these global maps of cellular structure. And so with that, next slide. So now Yue will describe the approach developed called MUSIC that was used to fuse these two large-scale data sets and create multi-scale maps of cell structure.
Thanks, Leah, for the great introduction. So I will briefly go through our method, which actually consists of only 3 steps. So the first step is how can we extract the information of the protein from the data modality? And our approach is by embedding each data into a low-dimensional space so that each protein gets now assigned, instead of an image or instead of an interaction network, it gets assigned an embedding from those data. So thus, an embedding is actually a low-dimensional representation of a complex data structure, which you probably most often see in the UMAP or T-SNP plots, if you've seen those in some single-cell RNA-seq studies. So in those cases, it's actually a very similar idea. So in those cases, in the single-cell RNA-seq study, you embed the complex transcriptome of the cell into a 2-dimensional space, in that case, 2 cells will be put together or close to each other in the UMAP or T-SNP if their transcriptome profile are similar. And here, similarly, we're embedding each protein into a 1024 dimension for image and for APMS separately. So the image embedding of the protein will then capture the global localization pattern of this protein inside the images in relative to the cellular landmarks that Leah previously mentioned, the cytoskeleton, the ER, and the nucleus. And on the other hand, the APMS embedding, it will capture the interaction neighborhood pattern of this protein within the protein-protein interaction network. So after we have this embedding, and if you think about in the T-SNP or UMAP sense, it's like kind of you're putting cell in a coordinate instead of 2D space, we're dealing with 1024 dimensional space. So it's hard to imagine, but it's still like you're assigning each protein a coordinate in this case. And then can we measure distance from them? And then we can maybe do clustering to find the protein communities. And so the next step we do is by feeding these embeddings, these features of the protein into a random forest regression to predict a general measure of pairwise protein distance, and exactly from which we then apply pan-resolution community detection to construct this multi-scale subcellular hierarchy. And this pan-resolution community detection, it's virtually similar actually to co-elution. So if you imagine that you have a column and you just input all the predicted pairwise protein distance, and then they'll say okay, let's now adjust the stringency of how these proteins, how stringent do we want these proteins to interact. So if you set a stringency really, really, really high, you say, okay, we only want protein pairs that are like 0 distance to each other, then in this extreme case, you're only able to find this kind of single proteins. But as you gradually relax the stringency, you're able to include interactions, first close interactions, which leads you to identification of these smaller protein complexes, but then gradually maybe transient interactions or distant interactions that leads you to finding larger and larger and larger protein communities. And how this is done computationally is here shown on the right. So you start from here, you start from bottom up, follow the arrows. So in the bottom here, you are setting the stringency really, really, really high so that in the network, you only allow edges that represent really, really close protein distances to appear. So in this way, the protein complexes or the connected protein communities you're able to find are those strongly connected or closely connected ones, and they usually form really, really small protein complexes. However, as you gradually relax the stringency, as you go up here, you're going to start to include edges that represent more distant protein distances or more transient interactions, for example. And here, for example, in the middle, you see like the red edges, they represent a larger distance than the yellow ones. And as you even go up, you lower the stringency even more, you then start to see those purple edges, which represent even larger protein-protein distances. And so this is how you're able to identify larger and larger protein communities that also contain the previously identified smaller ones. So by applying this to all pairwise protein distances that we have, so this is how we're able to construct the version 1.0 of our multi-scale integrated cell music, which is shown on the right. So you've probably seen a lot of protein-protein interaction network where the nodes represent individual protein and the edges represent the protein-protein interaction between them. So here we represent music similarly in the network form. However, the difference here is that the node, they represent, instead of proteins, individual proteins, they represent a protein community. So each node here contains a group of proteins that's assigned to this node. And the edge between the nodes represent the containment relationships between these communities. So for example, like cytoplasm and nucleus, they're the child system of cell, they're being contained by cell, and cell is thus their parent systems. And here, the size of each node represent how many distinct proteins are being assigned to this community. And also the color shows whether this has been previously documented or not, and thus allow us to distinguish between what are potentially just known systems and what are potentially putative novel systems that we can further dive into to see what we are able to learn biological insights from. And we found that almost half of our systems are potentially novel and might contain exciting biological insights. And furthermore, I'd like to stress on the advantage of doing this pan-resolution community detection instead of what you probably will often see as hierarchical clustering. So one major difference is we allow one protein system to be assigned to multiple parents. So through this way, this is a better representation of how you're able to capture the protein dynamics, for example, the pleiotropic of the protein and the multi-localization of the protein, and thus better represent the cellular biology. And so even though I'm showing like this as a more as a succinct network view where I don't really show the underlying proteins beneath them, but each node here, if you look on the left, is actually consisted of a protein protein-protein interaction network. So we know like which proteins are inside and what is the distance or what is the predicted distance between them. So this allows us to then be able to estimate a size or diameter of this cellular component. And this is how we're able to derive this size ladder shown on the right here. And also how we arrange each cellular component or each system in our music map in accordance to how large this system is. So how, like as I mentioned, size and how well do we capture? We compared what our estimate is to what the literature reports. So on the left is this comparison of 9, I think probably 9 or 10. I forgot the exact number, but maybe 10 cellular components that that overlap between music prediction and literature report. So on the x-axis is the predicted size or predicted diameter of these cellular components in music, and on the y-axis is the literature report sizes, the literature reported sizes of these cellular components. And so we can see that music accurately sizes biological systems across a wide range of physical scales. And on the right here is a cryo-EM model to further demonstrate that we do predict a size or diameter that's in accordance with experimental measurement, and also music captures the protein arrangement and the protein component arrangement within that is in agreement with the biological knowledge. So like from the beginning, we've been saying like we have two kinds of data, like we have image and APMS, but how do they actually like contribute to this final music hierarchy? So we assess this, like we assess the data importance by introducing by using a robustness metric. And so this is done by introducing perturbations into the input data so that you can observe like how does this perturbation from the input leads to the identification of or the recovery of the cellular systems. So on the left here, the color indicates the robustness of the system identification or system recovery if you're given both image and APMS data. And in the middle here is when you only give image data, and on the right here is when you only give the APMS data. So we can see that image robustly identifies cellular components that consist more of the upper structure in the cell, while APMS provides more information for the middle to lower scale in the cell map. And of course, you need both data to best recover the music systems. So here is on the y-axis is the number of music systems that is most robustly recovered based on the data that's being input. So what kind of novel biology can we get from this kind of cell map? So in order to assess this, we first did a global validation by performing 134 more APMS experiments with the help of the Bioplex team, and we validate this global validation help us validate 64% of music systems that they indeed enrich for novel protein-protein interactions. And also this enrichment spans across a variety of physical scales. So we also further dived into the biological insights that you're able to get from several of these stories. And today I'll be introducing mainly two of the stories, and for the rest, please do check out our paper for these biological insights. And so first, I'll be introducing the pre-ribosomal processing assembly, which is a novel protein assembly that we found really interesting in our map. So this assembly, which we named PRRPA, it consists of 7 proteins, and they're being grouped in the map due to the similarity in the protein localization, which is shown here as a nucleolar localization, and also due to their similarity in the protein neighborhood. However, do note that there's no APMS interaction among any of these 7 proteins in our input data. And here on the right, we show that we indeed observe a lot more new interactions. Actually, this is a very strongly connected protein assembly based on our new APMS experiments. We indeed recover more than half of about half of the interactions among these proteins. And this is due to, in the original input data, only one of the 7 protein, which is NBL, has been used as a bait protein, and leading to missing of the majority of the interactions that are actually here within this assembly. So in the new APMS experiment, by tagging 5 new proteins, which is labeled as dark blue here, as new baits, we successfully recover these new interactions. And so we suspect that this assembly, they participate in the pre-ribosomal RNA processing based on the known function of some of its members, and based on some putative ideas from high-throughput genetic screens, and also based on some pathology functions of the protein members. And so thus we tested like whether this assembly does participate in the hypothesized process. So we did this by knocking down each of the protein, and we do observe that knocking down each of them does perturb the mature ribosomal RNA, which is assessed by the 28S over 18S ratio here on the upper right. And we also did RNA immunoprecipitation qPCR assay, where you pull down each protein and see if it binds to the specific RNA species that you are detecting. And here we're detecting 45S pre-ribosomal RNA. And we do observe like these proteins, they do bind to the 45S pre-ribosomal RNA. So another very interesting story involves a protein RPS3A. So by name, you probably already guessed that it's known as a ribosomal assembly protein. And however, in our map, it is hanging out there with like 3 other RNA splicing regulators, SRM1, SNRP70, and U2F2. So this makes us wonder, like, maybe this protein has something other than its role as a RNA assembly, as a ribosomal assembly protein. So in order to test this protein as a potential RNA splicing regulator, we collaborated with our colleague here at UCSD, Dr. Gene Leah's group, and performed a ECLIP assay, enhanced UV cross-linking immunoprecipitation. And so briefly, for the ECLIP, you UV cross-link the cell to cross-link the interaction between the RNA binding protein with the RNA. And then by pulling down the RNA binding protein, you're able to detect which RNA species this protein binds to, and also where this protein binds to in the RNA. So this allows us to then be able to derive this kind of ECLIP profile for the RNA binding protein, and by characterizing like which region of the peak of this binding peak falls into among in the RNA, and here like we're categorized into like, for example, coding sequence 3'5' UTR, microRNA intron, for example. And we observed that RPS3A, the profile is highly similar to that of canonical splicing regulators and very, very different from canonical ribosomal proteins. And we further clustered the profile of RPS3A with all the publicly available ECLIP profiles, and we observed that RPS3A robustically cluster with RNA binding proteins that are specifically enriched for splicing regulation, further supporting the potential role of RPS3A in splicing. And that's like 2 of the stories that we have explored in our paper. And please do check out our paper for more details and more interesting stuff that we didn't mention in our talk. And going on, Leah will introduce you to like what we are up to now and what are the future potential directions for this work.
To summarize, for this first version of music just published in HEC 293, we developed this general approach that is able to systematically integrate these large-scale data sets from different data types. And this led to building a single unified map of cell structure, in this case from protein interaction and imaging data sets. And as Yue just talked about 2 of the stories, and was able to uncover novel biology and uncover novel subcellular systems that then we were able to go and validate the function of. Next slide. Looking forward, music version 1.0 was in the HEC 293 cell line. This was the cell line with a bioplex interaction network available at the time. And so although there were many proteins in the bioplex network, this cell line did not have quite so many images in the human protein atlas. So there's around 600 proteins in this first version of music. And on the right, we looked at how the subcellular annotation of each of the proteins for the red bars in music, so in HEC 293 versus the gray bars, so in all of Human Protein Atlas with the 13,000-ish different proteins. And so you can see it was like a representative snapshot of the cell, and but however, there are many more proteins that we would like to be including in these maps, and towards the goal of creating global maps of cell structure. And so now looking forward, the Bioplex team has now expanded to the U2OS cell line. So Ed Hudlin kindly sent us a U2OS protein interaction network, and this is the cell line with a very large number of human protein atlas images. So now we have around 5,000 proteins overlap. So we're expanding music to around 7 times the number of proteins in this more global expansion. So yeah, here's the very preliminary music version 2.0 consisting of close to 5,000 proteins in the U2OS cell line. And similarly to before, you kind of see, yeah, the top of the hierarchy, these larger subsystems factored into smaller and smaller subsystems. And yeah, next slide. And yeah, just looking through this preliminary map of cell structure, we were able to look and find, these many known protein complexes. And of these, like for example, these 3 were not in the original music map in HEC 293. So it's just showing that with this expanding this approach to U2OS and more globally, we're able to uncover more protein cellular subsystems. And kind of a similar idea is that Yue discussed before is many of these have bioplex interactions in the original network, but there's also many proteins that are brought into these subsystems that were not in the original bioplex network. So either through the network embeddings, so similar interaction neighborhoods, or through the image embeddings, we're able to, robustly create these cellular subsystems that we might have missed using just one data type alone. And so, looking even further forward, we're now looking to, explore this music map for novel subsystems and potentially design similar, functional assays to validate them. And then I saw a question in the chat that kind of addresses this. We're also interested in looking at integrating other data sets and other data types. So I just kind of want to drive home the point that this is, generalizable pipeline for integrating data. If you can create an embedding of your protein in a data source, then you can, integrate it into this pipeline. There's, it doesn't need to be limited to just protein interactions or images. And so, actually, we are looking at possibly integrating, co-elution, correlation profiling mass spec data, as well as proximity labeling data into this pipeline. And I hypothesize we'll get even, more robust and larger, more global hierarchies as we integrate more data. And finally, apart from, just kind of one final motivation for this work is creating these global maps of cell structure can give us, a better idea of disease. So, for example, in cancer, a commonly known problem with analyses is when you look at mutations at the gene level, although there might be, a small number of genes that are frequently mutated across different tumor samples from the same cancer type, the majority of genes will not be frequently mutated or recurrently mutated. So this paper here shown on the right, if we can use these multi-scale maps of cell structure to then look at the cellular subsystems that the proteins encoded by these genes participate in, then we can get an idea of recurrently mutated subsystems in the cell. So maybe either complexes or biological pathways that might be recurrently mutated across different tumor samples. Looking forward, just interested in using these maps to discover novel biology, but then also use to inform analysis of disease. And so, I think with that, you can chime into in this acknowledgement slide of how big of a team effort this was, and many different labs contributing, and definitely a big thank you to the IDACR laboratory, of course, and the Human Protein Atlas and Bioplex teams. And I don't know if Yue wants to say anything else, but thank you all so much for listening.
Excellent. Very interesting. So we do have a couple questions for us. You already answered the one, so I'll skip that one. First question, have you tried co-embedding the different modalities together? For example, like the CLIP model from OpenAI.
Yeah, that's actually another area like we've just started actively looking into is, definitely co-embedding these approaches. Right now I've been looking at taking the embeddings we already have and trying to, further low dimensionally embed those in the same embedding space, but potentially kind of, like the OpenAI, or I've been looking at Data2Vec and such. Trying to look at maybe starting at the original network and images and creating embeddings that way.
Excellent. Our next question, does the model benefit from the larger amount of training data that is available when integrating different data sources? Or is the data too heterogeneous, and would it still be better to create separate maps?
So I definitely think like the more data, the more it's going to be beneficial to the map, because like each data, it captures a different resolution or different snapshot of the cell, although like what resolution that data is to be determined. However, the more pieces we're able to gather into the pipeline, the more we are able to fully recover the cell structure. It does like get into a point, like for example, like when we integrate too many data sources, whether there's a point like the noises overlap or the noises like then like go over the signal, cover the signal. So it is possible potentially at that point, then we need to like start filtering out the data sources that is just too noisy to use. But at the current point, I think it's worth just integrating as many as we can find. However, like we do know that, for example, like when we develop this study, we want to restrict ourselves to only the HEC293 cell line, so that we at least avoid, especially in the data development phase, to like the noises that you're introducing when just integrating data from two different cellular backgrounds. So yeah, there are considerations, but if we're able to get as many data types from a same unified cellular background as possible, I do think it's worth integrating and generate a single map.
Yeah, and I think with the U2OS, we're still keeping it within the same cell, like two different data modalities coming from the same cell line. But as like we kind of use these as the, first steps for, like determining the best way to do these analyses. I'm also hoping we can expand to other, maybe more global interaction data sets might be available, or different data sets might be available that might not be context specific if possible. But we'll have to see.
Fair. That makes sense. So you mentioned filtering out the noise, the data sets that are too noisy. What kind of thresholds would you look at to determine too noisy versus, just chaotic data set?
Yeah, that's a really good question. It's definitely going to be an ongoing debate, like how do you define the noise in this context when you don't have a gold standard or a gold metric to assess it. I think potentially you can just see whether this data, like when you input alone, does it even contribute, for example, to the identification of the system. For example, like the one we are showing here. I feel like if like a data type couldn't even by itself like recover any of the system, then it's a sign that it's not useful and not worth integrating potentially.
All right. That's actually a really good metric. So we have another question here. Does the imaging data usually provide higher level information, for example, known structures, and act as a scaffold for the APMS data?
Great question. So actually, if you look here, it not only provide the upper structure, but it actually goes down. And also like a note that like you need both data type to actually best recover the systems. So even though the information or the influence of the image dials down as you go lower, it does still provide information, like it does still provide information of like which proteins should be grouped together. And also another very interesting finding in our paper is that using this kind of map, you're actually able to filter out interactions that are potentially more confident and biological meaningful, especially like in the sense of APMS. One complaint is that a lot of interactions are potentially just transient. You don't know what they're doing. They may be just like non-specific or just have no biological information. So by doing this, by integrating with image information, this is a way for us to also be able to identify those ones that maybe assign the lower p-value in the APMS, but still carry biological significance, and also identify the ones that, even though seems robust and strong, get a high p-value in the APMS, but potentially doesn't really have much biological meaning.
All right. So another question, what do you think is the biggest limitation at this point? Is it the number of proteins, the input data quality, or something different?
Yeah, great question. The limitation, I say, is still like the number of proteins, like because each of these, like we have been, a lot of group have been doing this kind of large scale effort. But like for example, when we first started this project, the HPA is not talking with Bioplex, so that is why the overlap is just so little. But I think especially as we're trying an effort here to like bridge the communication between these large efforts and to really dive into the value of having a shared set of proteins in the shared set of cellular backgrounds. I think we're gradually solving this limitation.
So related to that one, if you could, wish from a genie kind of, rules don't apply. If you could get a perfect data set that would fill in the biggest gaps that you want to fill in right now, what would that data set kind of look like? What do you think is the best one that you could get for that?
Wow, the perfect data set. Wow. I feel it's even a luxury to imagine that, but like in sense of perfect data set, I definitely want like well, if we go really bold, like all kinds of data from DNA all the way up to image, have that data for all the protein and for all the cell. And then like from that point on, it's really like a computational challenge. For example, like what is the limitation of the current pipeline? And this actually, we do feel like there needs to be also like a joint effort with, for example, physics group or just the informatics people that how do we then like best extract information. So this pan-resolution commute detection, there definitely needs effort, for example, from the physics group. And I think we do benefit a lot from their ongoing work, but if they can like have a better algorithm, we're definitely happy to use it. The trick here is that you want the systems to have overlap with each other, and you want the systems to be assigned to multiple parents. And even though our current approach does get us there, it's definitely not perfect, and there is a lot of limitation in the computational, and we're trying to solve it. For example, Leah could introduce like our previous effort in another method called high def. But there are also problems. So if people can join effort in thinking about like this unique kind of biological clustering, we're definitely happy to leverage, to bridge effort here.
Excellent. What about you, Leah? Any different kind of data set that you'd want?
Yeah, I was kind of thinking while you was talking the perfect type of data. Because yeah, I think what this kind of gets back to the earlier questions. What I find so interesting about this is in order for a protein interaction to happen, the proteins need to be in the same subcellular locations. That's what's kind of I find kind of like so cool about trying to use this imaging data in sort of a new way and also spatially place these protein interaction sets. I'm pretty interested in kind of what like the purpose of this community detection approach that Yue mentioned of allowing child systems to have multiple parents and vice versa. And so allowing for multi-localizing proteins, I'm pretty interested in, and how interactions between proteins change in different locations of the cell. So I think systematically like determining those kinds of things can be kind of difficult, but I'm pretty interested in how like proteins are participating in different complexes in different locations and what might be regulating that, such as different post-translational modifications or maybe different splicing variants at those different locations. So definitely integrating more mass spec and sequencing data too. So
interesting. So we have another, not a question, but more an ask for opinion. As someone doing method development for protein RNA/DNA crosslink mass spec, they wonder how much such a model profits from information on direct interactions. Also, for example, protein-protein crosslinking. What are your thoughts?
Yeah, I actually think we're interested, in looking at modeling within these subsystems. That's where, such things like protein crosslinking could, really help with actually like teasing apart subunits of complexes and, going even further, like an even higher resolution perhaps than we're like able to achieve right now with these more indirect methods. But I think definitely one day. I don't know if you has anything to add.
Yeah, definitely think these are all like important and important addition to music. Hopefully in the future that we have data to integrate and see how it contributes. And also like to second Leah's, like I think the especially the modeling, like this kind of like knowing which proteins stably interact with each other is definitely a great, very valuable addition to the current map, because like we really just tell you like what are the protein communities, but we don't know like whether they are stably or transiently interacting, unfortunately, at this stage. So this is where this kind of data could really help us.
All right. So give the audience a chance to write up their last few questions. While they're doing that, this may be entirely naive. I haven't done a lot of mass spec work, so I'm not very well versed in it. For me to set up a system like this, is there any kind of specialized knowledge would I need to be, really good at a specific Perl library, or would I have to have any kind of previous training, or would I be able to out of the box get it going, even though I'm might not know exactly where I'm taking it?
Yeah, great point. So we actually we actually spent a lot of effort trying to clean up the code, make it organized, and make it more. And we even read a very detailed step-by-step guide. So maybe it's all public on the GitHub, and also can be accessed via our portal page, or just like I think we also have the GitHub link pasted in the paper. So yeah, if anyone's interested, please try and let us know where we need to improve on making this more user-friendly. I say like currently we hope the only training or the only like prior knowledge one would need is Python. So from there, hopefully like our step-by-step guide and our code effort could just lead the user through like generating such a map from their data.